Okay.
So this is a bullet blender. We use this one for homogenizing some algae, breaking the algae, and extract hydrocarbon or extract protein from the algae.
Okay.
And this is, we have an instrument here that is called FPLC that we use for protein purification, like purified protein from bacteria, like such kind of things.
Okay.
And this is a cold room,
General.
which is maintained at 4 degrees. We store our plates, we store some chemicals and that kind of things.
Okay.
Sorry.
This is the cold room, that's okay? Okay, let's go.
Okay, let's go.
Now with the epoch. With the spectrophotometer also?
So here we have a spectrophotometer which we use for quantifying DNA and measuring the concentration of a DNA, RNA or protein samples.
Okay.
And then we have, this is the bench space where grad students work.
Okay.
We do research, we do design experiments, conduct experiments here. And we have another instrument called HPLC, high performance liquid chromatography, that we use for purification of peptides, purification of hydrocarbons, and you can use for so many other applications.
Okay. So let's go to the radioactivity.
Okay.
These are the chemicals. Okay.
And this is an area where we do experiments with the radioactive chemicals, like radioactive phosphorus, radioactive tritium, and that kind of things. As you can see, this place is surrounded by all these plastics, secured by plastics, and when we work with the radioactive chemicals, we work behind this shield and with the lab coat, gloves, and everything on it.
The waste also is there.
Yeah. And we put the waste inside that. We have a lot of this fume wood where we do experiment. When we get some waste from experiment, we keep it here.
Just
on these bottles.
Up the curtain, just to open it. Right. Okay.
Right now I don't have gloves because I'm not doing experiment.
Right. Okay, let's shake it. Okay.
And this is an area where we run our DNA gel. If you guys know what PCR means.
I think they are doing right.
When we run PCR and when we have to analyze our samples from PCR, then we run a DNA gel. This is the area for it. This is our office, grad student's office, or postdoc's office.
Coffee maker.
Everything.
Microwave.
So this is Dr. Edmondo's office.
Alejandra's office.
This is Dr. Alejandra's office, and undergrads and grad students' office.
Okay.
My office is on the other side.
Okay. So these guys, this is the pH meter for that.
Meter.
Balance. Okay. Spectrophotometer.
So this is a -80. As you can see, the temperature here, -79 degrees centigrade. We store our samples here. It's very cold.
Right.
Then we store our samples. As you can see, there's a lot of ice.
Right.
Here, and we store our samples.
Okay.
You can look at it.
Right.
And
that's okay.
These all things. And
okay.
This is a place where 4 degrees we store our bacterial plates. As you can see, some bacteria. Again, I do not have gloves.
Okay.
Because I'm not doing experiments.
What's that? It's a.
Oh, this is also HPLC. High performance liquid chromatography. This is a very old instrument.
Right.
We use it. It's really old.
Right.
Probably was made before I was born.
Right.
And we have -20. Again, this is used for storage of sample. The temperature is maintained at -20 degrees Celsius.
Yes.
And
okay.
Let's show you. We have another instrument here. This instrument is called gas chromatography, mass spectrometry. also called GCMS in a short form. This is a gas chromatographic part, and this is a mass spectrometry, and this is auto sampler, which comes here and takes the sample from this vial and injects here and analyzes the sample over the time.
Okay, very good. The control, these are the tanks of gases.
All the tanks of the gases. And this is another -80.
Right.
As you can see, the temperature is -80 °C, pretty cold.
Right.
Again, and here we have a cabinet where we store our clean flasks, beakers, bottles, and everything,
right.
medias, such and such. And this is an instrument where we use to take a picture of a DNA gel,
right,
or a protein gel.
Just open the shaker.
And this is a shaker where we grow bacteria, yeast. As you can see, right now I'm growing yeast.
Okay.
For some experiments.
Okay.
This is another centrifuge we have. This is a PCR machine.
Okay.
We have two different PCR machines. Let me open this and show you. This is a see there are 96 wells.
Right.
You can put 96 samples and do PCR at one time, and those are all the samples that was
okay.
For PCR. This is another PCR machine. We have microscope. If you want to see some pictures of the algae or bacteria or whatever,
right,
you can always check in this.
Another hood and the rotator.
This is another fume hood. We have a rotary evaporator where we concentrate our samples. Remove the solvent that we don't need to collect here, and the solvent goes to the waste here. Then the waste is designated for specific compounds.
Okay.
And this is another -20°C freeze.
Right.
We keep our enzymes, proteins here.
Right. Okay.
And then we have this block,
Uh-huh.
which holds temperature at -20, and when we have to transfer our protein samples and do experiment, we put it here and take it to our
Right.
bench and do the work.
Great.
This
Okay.
is another tank, nitrogen tank, and these all are outlets where we can concentrate our sample, using the nitrogen gas.
Great.
And please find out what nitrogen gas does. Why do we use for concentrating?
Okay.
And
I think that's
I think that's pretty much what we do.
Okay.
And
Thank you very much.
Ham
Tapa
See you
from
next week.
Nepal.
Yeah.
Okay. A PhD student, the most advanced, and he has been visiting Mexico several times.
Okay. Yes.
Thank you very much, Ham.
Thank
Bye.
you.